Welcome to the DNA Lab instruction video on home DNA sample collection for non-legal curiosity testing. Here are some important things to know. The kits are shipped out to you in discreet packaging, and our company name will not be on the outside of the shipping envelope. The person whose DNA is being collected is called the donor. Only use one collection kit for each donor. Additional kits are available upon request and are subject to additional fees. To maintain confidentiality, no names should appear on any samples that you submit. The shipping envelope will contain one or more individual collection kits, depending on the test that you ordered, a return mailing envelope with our mailing address and the file number, an information brochure with the paragraph about the child's biological mother highlighted for your knowledge, and an instruction sheet with step-by-step instructions on how to do the collection. For this example, there are a total of 3 collection kits because this is an example of a standard paternity test. One collection kit can be used for the mother, and one for the child, and one for the alleged father. The total number of kits that you end up receiving will depend on the specific test that you ordered. Before you begin the collection, please ensure the following. Rinse out your mouth with water prior to collection to remove any food that may be left behind. For babies being breastfed, their sample must be collected as long as possible after their last feeding. And we recommend a minimum of 1 hour from the time of their last feeding to ensure that the DNA collected from the baby is not contaminated by the DNA in the breast milk. Please avoid coughing, sneezing, and talking over any samples. Do not open another kit to collect a sample from a different donor until the first DNA collection has been completed and the sample envelope has been sealed. Each individual collection kit envelope will contain a sample envelope with an FTA card inside and a foam swab as well as a pair of disposable gloves. Before opening the collection kit envelope, it is recommended that you place a clean paper towel on your table so that you reduce the chances of any contamination from the surface used. Now open one collection kit envelope, put on the gloves, and retrieve the sample envelope containing the FTA card that you will deposit your sample on. Make sure you fill out the file number as well as the donor status, for example, mother, child, or alleged father, and the donor's ethnic origin. If your case involves collecting samples from multiple donors of the same donor status, please differentiate the samples accordingly. For example, if your case involves 2 different children, designate one child Child 1 and the other child Child 2 during each separate collection. These are the available ethnic databases. The ethnic origin that you provide on the sample envelope determines the database used for statistical calculations. If you choose not to provide the donor's ethnic origin, the Caucasian ethnic origin will be used as the default database, and this will be indicated on your report. Now take the FTA card out of the sample envelope. Write the file number and your donor status on the lines at the bottom of the FTA card. Be careful not to touch the pink area underneath the flap of the FTA card. Then use the foam swab to rub the inside of the donor's mouth for approximately 20 to 30 seconds. Make sure to rub both sides of the foam swab firmly against each cheek, as well as under the tongue, and behind the lip between the front teeth. Keep in mind that if you are too gentle with the swab, you may not collect enough cheek cells. Then press one side of the swab onto the pink circled area for 10 to 15 seconds and repeat for the other side of the swab. Here are some important reminders for when you transfer the swab sample onto the FTA card. First, make sure you press but do not rub each side of the swab onto the FTA card. Second, the pink circled area on the FTA card must change to a whiter color to indicate that the sample has been successfully transferred. If you see no color change, you can re-swab your mouth a second time and repress the FTA card. Third, we require cheek cells and not saliva for DNA testing, so the sample needs to be dry prior to returning the FTA card to the sample envelope. Note, if you have accidentally soaked the FTA card with saliva, you can place a clean tissue over the top of the card and leave it to dry before continuing to place the FTA card into the sample envelope. When ready, put the FTA card back into the sample envelope and then seal the envelope using the self-adhesive flap. After sealing the sample envelope, throw out any swabs, gloves, and any other disposables used for the sample collection that you just completed. Repeat the same steps discussed in this instruction video for each different donor, using a new collection kit each time. Once all samples are collected, sealed, and labeled properly, they can be put into the return mailing envelope. All sample envelopes for all of the donors go into the one return mailing envelope. Double-check that you have filled out all the applicable information for each sample envelope, and don't forget to fill out your own address on the top left corner of the return mailing envelope. The return mailing envelope with the samples needs to be sent back to the DNA lab, a division of MaryU NutraSciences. Please feel free to reach out to the DNA lab for any questions or concerns. And thank you for listening.